So we're continuing chapter 10 looking at muscle tissue, and I wanted to go back and go over some of the things that we already learned. We were talking about the actin molecule, which is the thin filament, and the myosin molecule, which is the thick filament. So I just wanted to go over how actin prepares to contract. And so in this picture here, we see actin, the actin molecule. Each one of these circles are actin molecules, and they're strung together. They're usually in two strings. And here you just see them in one string of actin molecules. But at rest, what we learned was that the tropomyosin molecule covers the active site on the actin molecule. And so when the muscle is at rest, the active site is covered up and nothing can bind to it. And then troponin is going to lock down that tropomyosin molecule over the active site on actin. So when a contraction starts, it's because calcium has been released from the terminal cisternae into the sarcoplasmic reticulum. The sarcoplasmic reticulum is a lace-type structure. It looks like a lace sleeve, I should say, over the myofilament, meaning over the thin and thick filaments, but it actually is a channel. And so the terminal cisternae is on either side of the T-tubule, where there's kind of a bulge, and there's a lot of calcium stored within that terminal cisternae. And when an action potential or a nerve impulse comes down the T-tubule, it will release the calcium into the sarcoplasmic reticulum. And from there, the calcium will diffuse out and flood over the top of those thin and thick filaments. So once that happens, calcium is flooding over the thin filament, and it can then bind to the troponin molecule. This changes the molecular shape of the tropomyosin-troponin interaction, and tropomyosin rolls away from the actin molecule. So as long as calcium is present, then the active sites will be exposed. And if the active sites are exposed, that means the myosin head can bind to them and we can get a contraction. So now we're looking at myosin, the thick filament. And I just want to describe to you how the myosin head moves. And ATP is what's required to move that myosin head. So let's just go through the steps of how the myosin head moves. So first of all, when the myosin molecule is inactivated, that means that the head has already been released and it's not in an activated position, it's in an inactivated position. So here we see the actin or the myosin molecule, there's the heads, it's been sprung already, and so it's in an inactive state. And so when it's bound to the actin molecule, an ATP molecule has to bind to it in order for it to be released. So here we have the myosin molecule with the heads in an inactivated state. They've already been sprung, and we have an ATP molecule that's attached to it. So to activate that myosin head, ATP has to split, and it splits into ADP and phosphate. Now, ADP and phosphate still stay attached to the myosin head, but the energy that was released in breaking that bond activates the myosin head or cocks it into an activated position. So now it's like a spring that's just ready to be sprung, okay? So ATP is now split into ADP and phosphate, and the myosin head is sprung or not sprung, but it's cocked. So in the next picture then, we see how the myosin head that is in the cocked position, in the activated position, it will just automatically bind to the active site on actin if that active site is exposed. So that's just what we're seeing right there. It's just a chemical attraction. It's going to happen. ATP is not involved with that. It just happens. Then in the next step, we see that two things are going to happen. The phosphate gets released from the myosin head, and that makes that bond tighter and stronger between the myosin head and the actin molecule. And then ADP gets released, and the breaking of that bond then is going to spring or pivot that myosin head. And as it pivots, it's moving that actin molecule with it because it's attached to it, so it's going to move it. Now where it's moving it to is over on this side where we have the M line. So it's moving that actin molecule closer to the M line. There's another actin molecule on the other side that will be moving towards the M line this way, so the whole sarcomere is going to shorten. Then in the next step, we see that there's now a space on the myosin molecule because phosphate left and ADP left, so there's a space there, and another ATP molecule, a different ATP molecule, will bind to the myosin head. And as soon as that happens, the myosin head releases the crossbridge from the actin molecule, and now we're back at step 1. So we go back up to step 1, and we see, okay, here's ATP. The myosin head's been released, and so ATP is going to, and the myosin head is already sprung, so then ATP is going to split again. It's going to split into ADP and phosphate, and we're just going to keep going down through these steps. So as long as ATP is present, and as long as calcium is present, we're going to be able to have this process occur, where the myosin head pulls that actin molecule closer and closer to the M line. So that whole process was called the sliding filament cycle, or it was called the contraction cycle, or it's called the Crossbridge cycle. So those are 3 names that are given to this process of those thin filaments sliding towards the center of that sarcomere. So in here we can actually see the sarcomere that goes from Z line to Z line, and that's where the blue myosin heads would be attaching to the thin actin filaments and pulling them closer towards the M line, and that would shorten the entire sarcomere. Okay, so that's called the sliding filament cycle, or the contraction cycle, or the crossbridge cycle. That's not the only process, though, that is necessary in order for a contraction to happen. The first thing we need is an impulse from a neuron. The neuron then transfers that nerve impulse into the muscle. The muscle carries that nerve impulse down to the terminal cisternae, and the terminal cisternae then releases calcium to flood over the top of the sarcomere. So there's 2 other processes involved. The first process that we're going to talk about is called the neuromuscular junction. It's what occurs between the neuron and the muscle. So we'll look at that process and we'll see how the nerve transmits its nerve impulse into the muscle. The second process occurs when that nerve impulse gets down to the terminal cisternae. And this process is called excitation, contraction, coupling. And so all this is doing is coupling the excitation from the neuron with the sliding filament theory, where we get the actual contraction. And so that's the second process that we're going to talk about. And the third process we're going to go back and review slightly is the sliding filament cycle, or contraction cycle, otherwise known as the Crossbridge cycle. So with all of these 3 processes, we then get a shortening of the muscle fiber. So let's take a look then at this first step, which is called the events at the neuromuscular junction. All right, here in this picture, what we're looking at is the axon terminal of a neuron. So that is the very end of the axon, and we see that it has a little bulge there, and inside that end, inside that synaptic terminal or axon terminal, we see these vesicles. Now these vesicles, these vesicles are filled with chemicals, and the chemicals are called neurotransmitters. The neurotransmitter that's used at all neuromuscular junctions is called acetylcholine. We can abbreviate acetylcholine as ACH. So you'll hear me talking about acetylcholine as ACH. So each one of these little vesicles contains thousands of those ACH molecules. So in the neuron, there's a nerve impulse that travels down the axon. And when that nerve impulse gets to the axon terminal or synaptic terminal, it causes the exocytosis of these vesicles. What that means is that the vesicles fuse to the membrane of the axon terminal and they release the acetylcholine out into the interstitial fluid. So there's a little bit of a space here between the axon terminal and the muscle. And that space we call the synaptic cleft. And when we look at the muscle side, wherever that neuron is coming in contact with it, wherever that axon terminal is coming in contact with the muscle, that space there on the muscle membrane is called the motor end plate. Okay, so acetylcholine gets released into this synaptic cleft, which is interstitial fluid. If we look at the motor end plate, we can see that there are these receptors over there. These are sodium-gated channels. And on each of the sodium-gated channels, they're chemical-gated channels, on each one of these there is a receptor. And that receptor is an acetylcholine receptor, which means acetylcholine can bind to it. So the acetylcholine molecules travel across the synaptic cleft and they bind to that acetylcholine receptor. And that opens up those chemical-gated sodium channels. Now we know that sodium is higher in concentration in the extracellular fluid than it is in the intracellular fluid. So sodium will diffuse in as soon as that gate opens up. So sodium diffuses into the cell. It's diffusing into the muscle cell. This alone with sodium, if enough sodium comes in and enough positive charges, because sodium is positive, if enough sodium comes in, that resting membrane potential of the muscle reaches threshold, we'll get an action potential on the side of the muscle. Now don't worry so much right now about understanding how sodium can lead to an action potential, because we're going to be going over that in another chapter. All you need to know though is that sodium enters into the cell and now we get an action potential. That action potential is going to travel into the T-tubules, which is filled with interstitial fluid, and it will travel down to the terminal cisternae and trigger the terminal cisternae to release calcium. So that's what happens with that new action potential that's been created on the muscle side. Now there's something else though that happens that's not going to occur indefinitely. We're not going to have acetylcholine being released indefinitely, so we're not going to have these sodium gates opened up indefinitely. As a matter of fact, the acetylcholine doesn't stay on that receptor for very long, maybe 20 milliseconds or so, and that's because there's also an enzyme floating around in that synaptic cleft that's called acetylcholinesterase. So it's acetylcholinesterase. And acetylcholinesterase is going to break down those acetylcholine molecules. It breaks it down into acetic acid and choline. Some of the acetic acid is going to be reabsorbed into the synaptic cleft so that it can be reused to make new acetylcholine. And then some of the acetylcholine is just going to diffuse away from the synaptic cleft, and then that won't even be used. So in any case, if we don't have a nerve impulse on the neuron, then we're going to stop the release of acetylcholine into the synaptic cleft, and then we're not going to get an action potential on the muscle, so then we're not going to get the release of calcium from the terminal cisternae. So if you understand how this process works, then you could understand how maybe some medications work. Like if we gave a person a drug that inhibited acetylcholinesterase, so that's the enzyme that breaks down acetylcholine. So if we inhibited acetylcholinesterase so that it didn't work, that means there would be nothing to break down acetylcholine, and we would end up with acetylcholine accumulating inside the synaptic cleft. That means that there's more acetylcholine to bind to the acetylcholine receptors, more sodium gates will be open, and the muscle will become more excitable, and it would be easier to produce a contraction. Another example of a disorder that happens here is called myasthenia gravis. In myasthenia gravis, several of the receptors, the acetylcholine receptors, are destroyed or damaged. So now there's no place for acetylcholine to even bind. That's going to make the muscle much less excitable. So we see with a person with myasthenia gravis, they're going to have poor muscle tone. We describe their facial expression as a mask-like expression because their muscles in their face don't have muscle tone. Because their receptors have been damaged. Okay, so now let's take a look at that action potential on the muscle side. So now we have an action potential on the muscle side, and that action potential is going to travel down the T-tubule until it gets to the terminal cisternae. Let's see what happens next. Here's the picture of the terminal cisternae, and a terminal cisternae is going to be found on either side of that T-tubule. Okay, so there's the T or transverse tubule, and we see the action potential coming down through that T-tubule, and it's going to hit the walls of that terminal cisternae. The terminal cisternae stores calcium. As soon as the action potential hits the terminal cisternae, calcium gets released, and it's going to go through those channels of the sarcoplasmic reticulum. Once in the sarcoplasmic reticulum, it's able to diffuse out of the sarcoplasmic reticulum, and it will flood over the top of the thin and thick filaments. So this is that second process I talked to you about, which is called the excitation, contraction, coupling process. Excitation, contraction, coupling. And what this means is that the excitation is that action potential that was started that's traveling down that T-tubule. The contraction means that the release of calcium is going to cause those thick and thin filaments to slide, creating shortening of the sarcomere. So this is kind of that intermediate step in between the neuromuscular junction and the sliding filament, right? So a couple of things that we're looking at here. First of all, what do you need in order for calcium to be released? You need an action potential. Okay, the action potential is a nerve impulse that's traveling down through that T-tubule. But then as long as the calcium is floating around, it's going to keep those active sites on the actin molecule exposed. As long as the sites are exposed, you're going to get a muscle contraction. And we know that you're not going to have muscles contracting all the time. They don't have enough of what they need to contract all the time. So calcium, as soon as that nerve impulse stops, calcium has to be moved back into the terminal cisternae. So calcium will be released from the terminal cisternae whenever there's an action potential signaling it to be released, and then once that action potential stops, then calcium will be pumped back into that terminal cisternae. So in order to pump that back into the terminal cisternae, we need to have ATP. ATP splits into ADP and phosphate. And when it splits, it releases a little bit of energy. And that energy then is used to pump the calcium back into the terminal cisternae. So an action potential will release calcium from the terminal cisternae, and when that action potential is done, then we need ATP to split or hydrolyze and pump that calcium back into the terminal cisternae. So there is a condition that's called malignant hyperthermia. Malignant hyperthermia. And so this means hyperthermia means high heat, malignant means very fatal or dangerous. So what happens in malignant hyperthermia is that the pumps that ATP activates to pump calcium back into the terminal cisternae, those pumps are not working, and it's a genetic condition. We see this when a person is coming out of anesthesia. The calcium pumps aren't working, so calcium doesn't get reabsorbed into the terminal cisternae, so calcium is present out in the cytosol of the cell. Calcium's bound to troponin, keeping that active site exposed, which means that the muscle will be in a perpetual state of contracting. When the muscle is continually contracting, it's going to produce a lot of heat. So that condition becomes serious when a person can't stop contracting and their body temperature rises to a dangerous level. Okay, but we're talking about normal conditions. So the action potential has traveled down that T-tubule. It hits the terminal cisternae. The terminal cisternae release the calcium into the sarcoplasmic reticulum. So now we're going to talk about the contraction cycle, otherwise known as the sliding filament cycle, otherwise known as the Crossbridge cycle. So here we have both the actin molecule and the myosin molecule, and we're going to put them both together. We're starting out where calcium is already bound to the troponin molecule and the active site on the actin molecule is already exposed. We're also starting out where the myosin head is already in the cocked position, meaning ATP already split, and now ADP and phosphate are attached to the myosin head, and there's this chemical attraction where the myosin head just attaches to the actin molecule, and that took no ATP at all. The attraction, the bonding there just happens even without ATP. So that's where we start. In the second step, then, we have phosphate leaving or breaking away from the myosin head, and that makes that bond between the actin molecule and the myosin molecule even stronger. Then ADP is released, and the breaking of that bond causes the myosin head to pivot. Since the myosin head is attached to the actin molecule then pivots, and now the actin molecule is getting closer to the M line. So here we can see how close the actin molecule was before the myosin head pivoted, and here we can see how close the actin molecule is to the M line after the myosin molecule pivoted. So the myosin head is still attached to that actin molecule, but another ATP molecule comes along and binds to the actin molecule, and that releases the actin molecule from the myosin molecule. So now they're separated. And we have the myosin molecule. It's in its inactivated state. It needs to be recocked in order to bind to the actin molecule again. So then we get to step 4. ATP splits, and it splits into ADP and phosphate, and that breaking of that bond recocks or activates that myosin molecule. So ATP splits or hydrolyzes, and the myosin head recocks. After that, we're going to start over again at step 1, where there's this chemical attraction. As soon as the head is recocked, and as long as the active sites are exposed, the myosin head will bind to the actin molecule again. Okay, so we just described all three processes. We described what happens at the neuromuscular junction, what the excitation contraction coupling process is, and then what this crossbridge cycle or sliding filament process is. So let's look at how the shortening of the sarcomeres can affect the muscle. When we were talking about the release of calcium from the terminal cisternae, and then that calcium gets released into the sarcoplasmic reticulum, which then floods over the thin molecules, it's important to note that if one terminal cisternae releases calcium, then all the terminal cisternae release calcium. So the entire muscle fiber then will shorten because all of the sarcomeres are going to shorten in that one muscle fiber. So it's an all or none type thing. Either all of the sarcomeres shorten or none of them are going to shorten. All right? So when a muscle fiber shortens, because all the sarcomeres are shortening, they produce what we call tension. And the next time we talk in part 3, we're going to talk more about what muscle tension is. And we're going to talk about other things that go along with muscle tension. So we'll see you next time.